Well, good morning.
Good morning.
I slept in today. This is awesome, and I had a big cup of coffee, so I'm raring to go. I did want to make an announcement that I got bored last night, and I changed the title of my talk. But it is the same exact talk. I just didn't like the title. I liked this one better. So don't fear if you're like, integrated pest management, that's not the right thing. You're in the right place. Okay, so we're going to get started. And I just want to mention that this work that I'm going to explain to you, I've been keeping bees this way, and maybe some of you have too, for, gosh, over 20 years, and been very sustainable, which we'll talk about what sustainable beekeeping means to me in my operation, and mostly my operation is for honey production. So I'm going to talk to you about kind of how I've done it and how the way I've kept my bees has led me to do a small research project with a wonderful student named Kathleen Evans. And I'm going to explain to you the results of that project. But of course, first I like to just show dramatic pictures because honeybees are awesome. And to give a little context behind Apis mellifera, because there are so many amazing species of Apis within the genus. So honey hunters, as you can see here, I was actually talking with Simon last night, who's a pretty good photographer himself, my goodness, about Apis dorsata and some of these other species of Apis. And you can see the early relationships with humans and honeybees is really similar to relationships we have with other wildlife. And I'm in the entomology and wildlife ecology department. And there's kind of this war between us entomologists and the wildlifers. And they're always like, oh, what we study is so important. I'm just like, what you study needs what I study to survive. All of what you study eats insects at some level. So get over it. You know, it's just kind of, I don't want to hear anymore. But I just was in Northern Ireland and I heard this most wonderful talk by John Lowry, I hope I'm pronouncing that right, about the spiritual connection between humans and honeybees. And it was marvelous, mind-blowing talk. And this really kind of shows you this picture. It's so surreal, but there's so many connections between humans and honeybees, from sustaining us with their honey and their proteinaceous pollen and larvae, which a lot of cultures eat, to just like understanding their unit, their social unit, and how they function as a superorganism. So they're very inspiring. And within the genus Apis, there's a lot of interesting bee members, and they have different ways of living together. There's about 10 recognized species, and Apis cerana, you might be familiar with, the Indian honeybee. Also Apis dorsata, pictured on the top here. Right here, this is Apis dorsata, beautiful bee, so photogenic. The coloration is just amazing. And this is Apis florea. And both of these honeybees actually make exposed nests. So it's this kind of exposed single comb layer. Whereas Apis cerana and also this one, Apis mellifera, which we're all really familiar with, the western honeybee, live in cavities. And so because of this different way of living, they have different defenses, they have different behaviors, which I'm not going to go into too much detail today. But I did want to go back to here and say, what's really interesting about the genus Apis is that all of these other species kind of live in the same range. Their native distribution overlaps, whereas Apis mellifera is separated, at least naturally, from the rest of the species. Humans have moved them all together and thereby kind of transmitting different pests and pathogens in the process. But originally, their ranges were separated, which is pretty interesting. Okay, and so their native range is Africa, Europe, and Central and Western Asia. So I'm always happy to get out of America because they're not even native there. And I even get trouble from other colleagues like, oh, you study an invasive species. And I'm like, do you like the food and the fruit on your table? You know, eat those words. I don't want to hear this awful gibberish. So if we look at the history of importation into the United States, it's really a rich history. Honeybee importation really began in the early 17th century. At least this is what we know from reports and records, written documents. But if we look further, the first ship's log where we can actually see that honeybees were transported was from the Virginia Company in 1621. And I always think about this, that long, dank journey for those colonies. Oh my gosh, it's amazing that any survived. And so I think about that a lot. And it was the dark bee of Western Europe, Apis mellifera mellifera. And I've heard I have to be careful about how I talk about this bee here to this crowd. But we still have pockets of Apis mellifera mellifera found in the US based on genetic analysis, which is a whole other area of research that I do that's really exciting, but I'm not going to talk about it right now. Okay, and you would think, and I often talk to people, you know, oh, they brought them over for pollination. Well, in 1600s, they did not understand the connection between insects and honeybees and pollination. That was an unknown. So they were bringing them over for very different reasons. They were bringing them over for honey, for the beeswax. And so it wasn't until later, about the mid-1700s, where this connection, this pollination connection, was discovered. So I find that so fascinating. Anyway, as we move on, the history of honeybee importation really kind of jumps back up about 200 years after that initial importation. And so 200 years after that initial arrival of Apis mellifera mellifera, there was about 7 to 8 additional subspecies that were known to be introduced into North America. And between 1859 and 1922, that's really the time period where they were searching. It wasn't that they were just bringing them over because they wanted honey and beeswax. Of course they did. But they wanted to improve the stocks in the Americas. They were seeking out good bees that had good honey production. They understood the pollination connection, so they wanted good pollinators. And they wanted nice bees, bees that they weren't scared to work and would chase off people. So it's really another wave of importation for a very different means. So this occupation of beekeeping in the United States and in North America really started to take off. And really, it was due to the fact that they're cavity nesters. Because they nest in a cavity, you can put them in the bottom of a ship, and you can transport them all over the world. You can move them into different orchards or different crops that are in bloom very easily. So quickly, you can take a colony of over 20,000 potential pollinators, flower visitors, and put it right smack dab of an orchard that's in bloom. And so because of this cavity nesting behavior, we were allowed to expand their native range, move them all over the world, and also select for favorable traits like honey production, where I live, overwintering, temperament. Those are just to name a few, and also managing for honey, which is very important to me. So if we talk about pollination, because arguably I think this is the most important ecosystem service that honeybees provide to us, it's such a simple and elegant process. This service, this kind of mediator that a honeybee serves as, this transfer of a plant's male reproductive cells, the pollen, to the female reproductive structures of a plant, the stigma. That's pollination. Very simple, very beautiful. And it's funny, I always give this question on my exam for my students, and they always get it wrong. I'm like, this is the easiest thing, but they're like, as soon as they put the word fertilization in there, I'm like, oh, I'm such a bad teacher. I'm going to retire. Because it's not. It's before fertilization. It's simply the transfer of the male gamete to the female structure. The fertilization occurs after. All right, so if we look at the staggering statistics, because some people, unlike you, who already know the importance of honeybees, don't know the importance, and so I have to smack them in the face with statistics. And globally, insect pollination services are valued at about $215 billion a year, which is a gross underestimate. I don't even know how they come up with these figures. It's kind of ridiculous, but all I can say is they're really important, right? So bottom line. And if we look also at the number of crops, it's important for pollinating more than 75% of crop species. And right now, sometimes I worry about just singling out honeybees because there's so many important pollinators. And sometimes honeybees aren't the best pollinators, so I have to give props to other pollinators, like megacylids you see there with the pollen on its abdomen. This is where I do need the pointer. You can see the pollen on this abdomen of this megachylid here. Of course, many different Lepidoptera, butterflies, and moths are important pollinators, as well as this is Pepinapis pruinosa, which is the squash bee. It's an obligate for squash. Squash flowers need this bee. They're specially designed to carry the pollen. They sleep and mate and go on dates in the bloom. That's cool. You want to go flower dating? You know, as really interesting stuff. Also syrphid flies, bee mimics. Here, I'm sure you've seen many of these. I actually saw one when I went to the Giant's Causeway. It was on a bramble. I was like, a syrphid, you know, a UK syrphid. That's so cool. And so also these weird apples with facial hair. We don't have those there, but I thought that picture was so disturbing, I thought I would disturb you all this morning with it. All right, and also the staggering statistics on a global scale of just the ecosystem services it provides for all organisms. Okay? Enables reproduction in 94% of flowering plants. Foundation for the promotion of biodiversity. So not only does it allow plants to expand across the landscape, which many other organisms need to survive, if you were to take away this pollination service, our food networks and chains and webs, however you want to talk about them, would collapse. So it's pretty amazing. In the US, the statistics, these are what they've come up with, 47.1 billion every year, of which 17 billion around is honeybee pollination, and the rest is other bee and other pollinator, other flower visitor services. So you can see the importance just in my country. I'm sure some of you have seen this. I think this is really clever marketing. I'm kind of obsessed with marketing and the creativity of humans to get their agenda across to others, and I really like this. You can see your produce choices with bees on the top, your produce choices without bees on the bottom here. And Whole Foods, which is a market in the US, this was their marketing campaign. They linked up with a bunch of different honeybee associations and did a really great job of kind of affecting the general consumer base. Also, I'm sure you've seen this, breakfast with honeybees, breakfast without honeybees. And my goodness, that says it all right there. Once you show these very simple pictures that really can relate to any person who's looking at them, and they can start to see, okay, yeah, honeybees are pretty important. So that brings me to population declines in honeybees in the United States. I'm sure you've heard the media's just gone crazy with it. And a lot of the statistics are based on commercial beekeepers. Because honestly, the small-scale beekeepers, and even myself, I don't like surveys. So I'm actually probably pretty lame for not filling out these surveys and lending to the general knowledge. But sometimes I'm just too busy. And also I'm just like, how are they crunching these numbers? So I'm very skeptical. But the Bee Informed Partnership has found and followed losses every year, really focusing on overwintering. So after winter, they're coming out of winter. What are the losses? Between 2012 and 2013, there were colony failures between 40 and 50%. This past year, they weren't quite that high, but they were still pretty high. Not sustainable. If that was really capturing honeybee population loss in the US, then it wouldn't be sustainable. However, I don't think it truly captures it. Now, so what is the problem? Why are we having these losses? Well, this paper came out, and I think it's just fabulous. You can see this is the citation here for those of you who want to jot that down, and I have a feeling this is recorded, so it'll probably be available on there. But this slide here basically shows you the interaction of many different things affecting honeybees. It's not one thing. It's the synergism between all of these things that in varying degrees, depending on where the bees live, what the land use is around your apiary, et cetera. So pests and pathogens, which also, coupled with intensified land use and pesticides, coupled with phenological shifts, different flowering times and bloom times that the bees might be not triggered to. Also stress, especially if you think about the large commercial operations that move their bees around on semis. That can't be very peaceful living. Going from monoculture to monoculture, as well as invasive species. So the loss, and this is more true for native bee populations. But you can see all of these things together. Really, it's amazing how resilient bees are when I think about it. So this talk is really going to focus on pests and pathogens, and I really am going to highlight varroa, because that's probably the biggest concern I have in my apiary. And you can see here, I have a whole host of different pests and pathogens that are common in the US and where I am. Varroa mites, nosema, small hive beetle, wax moths, American foul brood. But the varroa mite is a different type of mite. It is very difficult to deal with because it lives most of its life, either reproductive or phoretic, on the bee. So in order to kill it, it's tricky. It's tricky stuff. It's an ectoparasitic brood mite. You can see it up there on the corner on this pupae that's been pulled out so that you can see the mite for a nice photo op. And then you can see it at its phoretic stage here on the adult bee. And so sometimes they'll hide in between the sclerites too. They're, just so they don't get groomed off. So that's kind of where they can occur. And they also vector viruses. And I talked a little bit about viruses yesterday. And what's so difficult with viruses is that if you're not really trained and have connection with your colony to know how they're kind of doing at a deeper level, how they're functioning as a colony unit, there's not a whole lot of signs, often until it's too late, and the colony's already in a downward spiral or trajectory. And so this is a really interesting graph or map showing you the global distribution of varroa mites starting in 1912. And you'll see you'll be able to look at the year up at the top here, okay? So you can see here how the varroa mites are spreading globally. And pretty amazing how quickly this mite just traversed oceans, just this global successful spread of this very, very hard to manage parasite. So I believe it only goes up to, 1992 maybe? Yes. Or no, 93, 97. So I think that's very visually interesting on many levels. And the life cycle of the varroa mite, oftentimes you think about, okay, its true host, Apis cerana. They've been able to co-evolve with this mite over years and centuries. And so they can deal with this mite. They can live with this mite around without it tanking the colony, without complete destruction of the colony. And some differences between Apis mellifera versus Apis cerana is found in the life cycle of those bees themselves. You can see here the queen in Apis mellifera, 14 days, 7.5 of the days are capped as an immature. And worker 21 days, the drone 24 days. And the main thing I want you to see and notice the difference of is the worker. In Apis cerana, the worker is 21 days, 11 of which are capped. In Apis mellifera, the worker is 21 days, 12 of which are capped. Okay? And drones have 14.5 capped days. It's this length of being capped that allows that mite to go from just hanging out and getting a sip of blood or hemolymph to reproducing. Okay? So that longer capped period in Apis mellifera allows the mite to become reproductive. Not good. And you can see why they prefer drones, because then they have this extra long time period in this cap stage where they can just have a mite orgy. Sorry if that's a little too much for this morning. It's a bad visual, I know, but it's true. And so oftentimes people have tried to kind of figure out how this would break down. 2.6 female offspring, that's mites, female offspring mites per drone cell during that capping stage, and 1.3 female mites offspring per worker cell. Okay, so now you can start to see the reproductive value and fecundity of these mites in Apis mellifera. So Varroa destructor has really changed beekeeping and the art of beekeeping. I think more so the art, the enjoyment of going in there and experiencing your bees. When you see them sick and you see the deformed wings and when you see the mites on there sucking on them, it's disturbing. And it's really changed it forever. So the goals of this project were to begin to develop an integrated pest management strategy for hobbyist beekeepers to control varroa mites. Because we did a survey. Look, I did a survey. I'm not even good at doing them myself, but I made other people do a survey. And their number one concern was queens and queen productivity, but also varroa mites. And all of them wanted a non-chemical solution. Don't we all, right? And so really what I wanted to do, because I don't treat in my hives, because I produce honey, and I don't think those two go together, was to test the efficacy of splitting, so splitting my colonies, in reducing mite populations and varroa vectored viruses. And so hoping that by using this technique of splitting, I would be able to not get rid of all losses. I'm going to take some losses. I always take some losses. But with this splitting, I can rebuild and keep a sustainable number of colonies at all times. That is my goal as a small-scale beekeeper. So if we look at the integrated pest management program, it really was coined, this term, in 1972. And it's an approach that combines all things: chemicals, biocontrols, cultural, mechanical. And so it doesn't exclude chemicals, but it makes it so chemicals is the last kind of option that you would choose. And by combining all of these other options, hopefully you can reduce the overall amount of chemicals into the system that you're trying to control the pest in. Very simple, okay? So it's really kind of interesting to look at the history. I didn't realize how awful my font color choice was. I apologize. That says, prior to 1939 is the pre-insecticide era. In the 1930s, the trend was towards synthetic compounds, DDT and organophosphates. Post-World War II sparked the use of DDT for agricultural pests. So you can kind of see this ramping up of these chemicals in the ag arena. And so IPM, they were starting to see, after they used these chemicals for a certain amount of time, they were losing all the beneficial insects in these systems. And these ecosystems were starting to become unbalanced. And they were losing crop yield even when they were reducing pests. And sometimes the pests would come back because they became resistant. So IPM was in direct response to the overuse and the effects of purely using just chemicals. You guys probably already know all this. So what did that lead? It created this super bug. I don't know if you've seen this guy flying around, but it's pretty amazing. And the key to successful IPM is to know when there is a problem, to be able to identify when something's not quite right, like a giant varroa mite. I took this picture in Devon 30 years ago. Awesome. I wanted to take that mite home with me so bad. Oh. Anyway, so we need to figure out when control is needed. And in crops, the presence of just a few insects probably isn't going to have an effect on crop yield. And I know this is kind of general pest management in fields, but you can think about it and transverse it in your mind to your colony, okay? So pest control costs money. Miticides, all of these different things, traps that we buy, all of it costs money. All of the things we buy. It's only worth controlling the pest, actually purchasing these different things, traps, chemicals, whatever it is, if the money lost in yield is greater than the money spent controlling it. Unless you're weird and obsessed beekeepers like we are, and we don't care, and we lose out money every year probably, taking care of our bees. But if we look at this kind of general terminology, classic terminology associated with IPM, the pest population at which the potential loss of yield equals the cost of control is known as the economic injury level. This is when it pays to spray. And all big farmers, even small farmers, classically have followed this model. And they'll monitor their fields or their hives for pests to find this economic injury level. Okay? So it's determined through research on effects of insects at different populations on crop yield. You can see this poor cabbage. I think it pays to spray. Or do something. My gosh. So if we look at another term, this is the economic threshold. This is the action threshold. And this threshold is set lower than this economic injury level. So there's time for the treatment that you're going to do to take effect. So you don't do it too late, right? And so when you should take action to prevent increasing population from reaching that economic injury level. And those are soybean aphids on a bean leaf there. You can see pretty high density of pests. And to put this into a graphic, you can see here, this is your pest population, this red line here, okay? This is that economic threshold. So when your pest population, if you're out in the field or out in your colony doing sticky boards or whatever type of monitoring, once you hit this economic threshold, the pest population hits there, that's when you would want to do some type of treatment, not necessarily chemical. There's a whole toolbox available of things that you can choose to deal with at this economic threshold. Okay? And so the idea is if you treat here at this economic threshold, it's going to bring your pest population down. If you don't treat at the economic threshold and you get to the economic injury level, then it might be too late. Okay? And so this is the pest population without control. And so above the EIL, the benefit outweighs the cost. So that's kind of simple. And so this integrated pest management approach, the goal is to keep the population of pests below the EIL. Not to wipe them out completely, because that's just never happens, right? But to use all different tools in your toolbox to try to keep them below that EIL. And so in order to know if you're keeping them below the EIL, you have to monitor. There's all different types of monitoring depending on the pest. It could be simply just hiring a bunch of undergraduates who work very cheaply and putting them out in the field and making them scout. Also, it depends on the EIL, the pest itself, and the crop that you're dealing with. Monitoring for us would be using sticky boards to pull out pupae and actually count the number of mites on purple-eyed pupae, which is very laborious, but very important measure of the reproductive level of mites in your colony, which is a much better estimate of how the mites are truly affecting that colony than the phoretic sticky board. All right, so IPM, you want to base decisions on economics and scouting data and try the most vulnerable aspect of the pest. So that's important when you need to understand the life history of the pest. When's the best time to attack it? For the small hive beetle I talked about yesterday, it's at the pupal stage because it's outside of the hive and it's vulnerable. And so you just want to kill those pupae, right? And so each critter has its own vulnerable time when it's best to attack. I showed this picture yesterday. This is great. This is a beekeeper IPM pyramid, which I think this is just really great. You can see specific to beekeeper and IPM, the different cultural controls, the physical and mechanical controls as you go up the triangle here, biological control, chemical. And as you go up, it comes from prevention to intervention. So your involvement in controlling the pest increases, as well as the toxicity of your management option. But this is a great pyramid to kind of refer to when you're trying to look at different ways to control pests. What are the tools in your toolbox? So when do we need an IPM approach? Well, I would argue now. If we look at the chemicals, there's pyrethroids, the taufluvalinate or apistan, basically does nothing. Resistance was actually seen in the United States 5 years after it was registered for use. And as well, we see also resistance, complete resistance to coumaphos. And so we get about 5, maybe a little bit more years of use of these chemicals before they no longer work. And now studies have shown that a lot of these compounds, of course, have sublethal effects on the honeybees themselves. Because as I said yesterday, you're trying to treat an arthropod, or kill, not treat, kill an arthropod on an arthropod. All right, so additional miticides, we've tried Mitaway 2, different formulations of formic acid with scary results, sometimes to the colony, to the queen's laying, to the actual applicator. Apolife VAR, all different types of thymol blends, essential oil blends, also menthols. Apiguard, I don't have them all up here, but you know what's available to you and what's registered here for you guys to be able to use. And there's pros and cons. Varroa are constantly building resistance. The pros for some of these at first might be reduced mites, but is that sustainable? Is that going to be forever? No. The cons are some of these are only effective at certain temperatures. They volatilize out differently depending on the weather. Also, it can affect queen and drone development. Also, residues of these different things in the honey. One of the top residues in wax comb in the US is taufluvalinate and its metabolites, and coumaphos and its metabolites. So we are basically toxifying the house that the bees are living in and that their larvae are in. I mean, I'm not blaming anyone. I'm frustrated. You know, this is frustrating stuff. So how do we deal with all this? So what could be some possible components of an IPM program for varroa? And I'd love to talk with some of you later and hear what your ideas are, because I think it's going to take a collaborative mind to figure this out. But genetics, there's a lot of great genetic lines that are being bred. I would love to hear about some of the lines here. I can talk to you about some of the ones that I know are successful in the US. We have varroa-sensitive hygiene. So this would be VSH behavior. You can't really see it well, but there's the star-line stock that's maintained by some still in the US. Russian honeybees are, it's like a mafia over in the US. The Russian honeybees, it's just like, do you like Russian? Yes, I do. They're great, you know. But actually, I was involved with Tom Rinder, who brought over some of the early lines with a Russian bee breeding project, and some of them are wonderful. And I think, but some of them are really bad in the early stages, but they very much controlled the breeding of those. And so some of them are pretty good. The Minnesota hygienic, there's others too. This is just a few that I'm going to mention. For our project, for our purposes, we chose to use bees that had varroa-sensitive hygiene trait. And this picture here kind of shows you generally what that trait is. This is controls. We've killed the brood with some liquid nitrogen right here, and on both sides. And then we go in, and 24 hours later, 48 hours later, we actually see what's been removed. And what they'll do is they'll uncap a bee that's in the pupal stage, in the cap stage. You can see they remove the wax, and they're pulling out this pupae, okay? Because they can detect that there's something awry underneath the capping. And I'm sure you've seen this trait here. It's all over the US. In my yard, it's hard sometimes, because sometimes I'm wondering if I have inbreeding going on because I have all these holes in my brood pattern. And so how do you distinguish between this VSH trait and, you know, diploid drones, you know, from inbreeding? It's tricky business sometimes. But this is the trait we decided to use, this VSH trait. Here's another picture. Actually, I got 3 years ago when I visited Bishop Waltham and I spoke at the Meridian Beekeepers. And this was debris on the bottom of their hives. And you can see this area right here, a lot of these mites had bites taken right out of them. And also all these pupal parts on the bottom. So this VSH trait was definitely being expressed with also aggressive mite biting behavior in that particular colony. And so these are some of the behaviors that genetic lines are really focusing on. All right, and also in terms of management and pest monitoring, how do we manage and really understand the levels of varroa in our colonies? Well, there's a lot of different mite monitoring techniques. There's sticky boards, powdered sugar, there's beetle blasters to understand beetle levels, there's drone brood removal as well. So you can actually use the drone comb. Once it's fully capped, you can take it and not just freeze it, but you can get a good idea of the reproductive potential of the mites in your colony by pulling out those drones and counting the mites in the cells. So there's all of these different monitoring techniques that we can use to aid us in understanding the levels. And finally, the most important part of the IPM program that we tested, along with this VSH trait, and also some of these other monitoring techniques, was the use of splitting. And really, this idea that swarming has been shown that, especially with feral colonies, to lead to a stable host-parasite relationship between honeybees and varroa mites. And I know Tom Seeley has talked to this crowd before. Me and him do some work on the Arnott forest bees, because we're both very interested in how bees live in nature without humans getting in the way, right? And so this idea of using this break in the brood cycle, which varroa mites are completely tied to, they need brood in order to reproduce. How can we use this break in the brood cycle in order to sustainably keep our colonies alive and dying from varroa? So for those of you who are new to beekeeping or newer, you have a parental colony, and with a split or a swarm, what happens is the old queen and more than half of the colony, I know some of you just heard Juliana's talk, so you probably got a wonderful education on this topic, will actually leave the parent colony and start a daughter colony somewhere else, leaving in that parent colony a new queen to be raised and mated, and etc. So there's a break in the brood cycle because that new queen in the old colony has to actually get mated, come back and start laying. So you have this time where there's no brood. So varroa can't increase, right? And then in the split or the swarm, depending on, let's just talk about a swarm here. That swarm, once they land, they might have to build comb, etc. And so the queen, there's also a break before she starts laying, and there's brood at the stage that it needs to be for varroa. So this is kind of the idea behind this break in the brood cycle. Okay, so it was really important because we actually did a citizen science program with this, where we had beekeepers doing the same things we were doing, so we could increase our sample sizes, and also just for fun. And so a lot of them had to be taught swarming signs. What are signs of swarming? And so delay in laying, the presence of queen cups, congregation outside the hive. That might be too late. But you can see, oops, them flying away. So there was a lot of education in this project, which was great. And in this project, we had two different apiaries. We had a splitting apiary, and we had a swarming apiary. The splitting apiary was Langstroth hives, a lot of them, and I'll show you the experimental design there in a minute. The swarming apiary was my teaching apiary. And what's great about that apiary is that all the colonies are surrounded by really tall trees in the middle of fields of corn. Well, that's not so great. But what's awesome is that if it swarms, they just go straight up. And so if I scout every day, I catch all of my swarms because they just go straight up. And so I have a very good swarm capture rate in my apiary. And so this allows me to be able to actually look and follow the parent and then also the new daughter colony or the swarm and track them over time. So the design in the splitting apiary, you can see here we had 20 original colonies, and so I'll try not to confuse myself or you. 10 of them were designated as control colonies, and 10 were treatment. In the 10 control colonies, we did swarm prevention, and we did not split. In the 10 treatment, we split the colonies. And what you would see is you'd have the 12 control colonies, and then you would have 12 parents and 12 daughters, or 10 parents, and 10 daughters. Okay, so this is basically how the design looked. So we would follow the controls and look at varroa. We looked at bee population growth. We looked at honey production. We followed all the colony productivity measures that we could in both of these sets. In the control colonies that were not split, and then in the colonies that both parent and daughter, so the split group. And we compared the differences to see if there was any difference. What did the splitting do? Okay. Also, we did drone comb removal. We were interested in drone comb removal in combination with splitting. So by using drone comb removal early in the season, we did an early spring drone comb removal, and then for half of the colonies, and then in the bottom half, we didn't do anything. So we were really interested, because the literature is still pretty, there's some say that it works, but not forever. It doesn't work it doesn't keep them down for a long period of time. And then there's other work that shows a slight significance in a drop of varroa if using drone comb. So it was just something that we thought we'd throw in and look at. The protocol, the drone comb removal time periods, we did early spring and none. We used the VSH bees, the hygienic behavior bees. We had the treatment colonies, parental and daughter colonies, and then the control colonies, where we used swarm preventative tactics, killing of queen cells, clipping of queen's wings, which really does nothing, and monthly recordings of colony health. Okay. We also, for monthly recordings, we looked at adult and bee and brood populations, so we went through frame by frame and recorded everything. We looked at brood health, hive aggression, foul brood, queen cells, presence of chalk brood, mite monitoring. We pulled pupae, purple-eyed pupae, to look at reproductive mites, as well as sticky boards, so phoretic mites. And we did samples that we took straight from the hive where we looked at nosema and looked at virus loads. The mite monitoring techniques that we used for phoretic mites, we did 60 mite threshold. This is a special sticky board that was developed by Penn State. It's like a game, really. It's like multiply the mites in the black squares and subtract them by the white, it's this. But a lot of people use those. We did this sugar shake, and then we did these sticky boards, which is a 120 mite threshold for 3 days. This is what our data sheet looked like, because we had to make it quite easy for participating beekeepers to use and understand. You get that? I still think it's confusing. And so this is a test to wake us up a bit. I'm going to show you a brood pattern. For our brood pattern, we decided to have a scale of 1 to 3, 3 being the strongest, the best, and 1 being the weakest. I want you to shout out what you think. I hear 3. I hear 2. 3. You twos must have some pretty nice brood patterns. Okay, what about this?
1.
It's pretty bad. Yeah, you guys all got that right. 1 and a 2, I hear. 2. So you can see it's pretty subjective, isn't it? Which makes for some noise in the data, but that's the best that we can do. And we also looked at populations, and I'm going to walk you through how we did this visually. So if we look at here, we divided each side of the frame into 4 quadrants. And with this, we would look and say, okay, we'd smush when we were looking at bees, we'd smush all the bees in our brain into one of these quadrants. So if we took all those bees, it would make about 1/4 bees for that particular side of the frame. And about 3/4 brood, if we were to smush all the brood in there, pretty close to 3/4. 1/4 pollen, and 0 honey. There's some nectar in there, but not enough to really say much about anything. And there's a little bit of nectar, but it doesn't make a full quadrant, so we just put a check that there's nectar present. So that's kind of just to give you some insight on how we get our colony productivity assessments. Maybe that's similar to how you guys do it here. And then, so this would just be doing it again. I'm just going to quickly kind of go through here. Dronage. My student came, I don't know if you guys call drones that, but she's always like, dronage. Very weird. It could be a new word. We could start a trend right here at the honey show. Okay. And so really, just inspired by how bees live in nature and this idea that they can coexist with their parasites. And how do they do that? It's amazing to me when we think of bees, it's really hard for us to understand how they're doing it without us. It's pretty interesting. So wonderful work done by Tom Seeley and Ingemar. And this should be inspirational to us, this idea of these things being able to coexist. We're not going to eradicate varroa, but we can keep it so our bees aren't going to be completely killed and unproductive. So possible factors that may allow coexistence in nest in nature of these two things, and Tom has a great talk which talks about nest densities, actually the density of colonies in an area, and how we as beekeepers cram them into an apiary, which induces robbing and drifting, whereas in nature they're much more spread apart. Also nest size, the actual size of the colony itself. I see people and they're like, oh, this is my colony, it has 3 supers and 10, 3 brood chambers and 10 supers. I'm like, oh, how are you going to overwinter that? How are you going to consolidate that down? Like, is bigger better? I probably don't think so. They have to thermoregulate that whole, in certain times, if the weather and the nectar's there, but other times they have to thermoregulate that whole entire beast of a colony. So we have to think about what's the best nest size and sex ratio, drones to workers and queens. Also swarming. In nature, that's the way they reproduce at the colony level. This is a natural reproductive phenomena for honeybees. and we oftentimes prevent them from doing this. Reduce large-scale movement of bees, so actually moving them around from different apiaries to different sites. And this is the population dynamics of honeybees and mites. This is classic. This is was used in an article in 2012 in PLOS One. But I love this diagram because it really explains what's going on where I live. In January, you can see here are the bees. We still have this downward trend in adult bees here. And right about April, depending on the weather, you'll see this kind of increase in brood and also bees. What's triggering that? The nectar flow, pollen. It's all environmental cues. It's this feedback loop telling that colony, food's around, let's start building up, right? And so you see this upward trajectory in the bees and in the brood. And slowly, these devilish mites that need the brood are tracking the brood and starting to reproduce and get more and more. And just when the brood starts to decrease because the temperatures drop and the bees start to decrease because the queen has stopped laying, you have your winter bees in there now, you start to get your highest level of mites. Bad timing, going right into winter. So timing, that could not be worse. So the idea here is, okay, if you're splitting a colony, when do you split? When do you split so you still have enough bees to take advantage of the spring nectar flow, which is the biggest nectar flow where I live, but you can still stop that upward climb of varroa mites going into the winter? That is the magic question. And so we designed a project in 2012 where we did early splits. Okay? And the data that I'm showing you here is doing splits, and you can see we did the splits in June. Okay? So we have, you can't see it there on the side. I wonder if I don't think I can make it any better for you. But these are our control colonies, and these are our treatment colonies right here. Okay? And so in June of 2012, we got a late start that year, we split our colonies. And what you see here, these are the control colonies, these are the parent colonies, and these are the daughter colonies. Okay? And already with bees, which makes sense, you're seeing a significant difference in adult bees between the control colonies and the treatment colonies that you've now split, because you've literally split them so they have less bees in each one. And you can see that this effect on adult bee population continued into August. Down here is June, July, August. So you can see here, of course, it makes sense, by splitting the colonies, you are getting a reduction, a significant reduction in adult bees in the parent colony. Now, if we look at brood area, this was an interesting revelation to me. And I wanted to look at this more. There wasn't any significant difference in the amount of brood between the control colonies and the split colonies. Actually, the the daughter and the parental colonies built their brood right back up. It wasn't long. They rebounded quite quickly from being split. And if we look at the mites, between our control, daughter, and parent, you can see here in June, right when we hadn't done it quite yet, we just have the control and the treatment colonies, they started off pretty equal with the amount of mites. And then as we move to July, you can see already between the daughter and the control, we're seeing a reduction in mites, but there isn't a significant difference between the control and the parent. However, once you got into August, which is more of a critical time where usually you have higher levels of mites, we do see a significant difference between the control colonies and the treatment colonies that were split. So this was, a good sign. Maybe we should look into this further, even though this is how I always run my colonies. I split a lot to break a brood cycle. We decided to do the same thing in 2013 with more colonies and to actually do a full May through October. And you can see here, again, in May, we split in May. And so you can see the difference here in our parent colony in the dark, the control colony in the light gray, the daughter colony here, and already in bee population, you could start to see these differences. You can also see them in July, the control colony, of course, having more adult bees than the ones that were split. And then, however, you start to see them all leveling out going into October. If we look at the varroa population over the same time, you can see here the ones with the stars, and same with the previous slide, are the ones where we saw significance. So the actual numbers were significantly significant based on statistical analysis. You can see in July, August, and September, we're seeing a significant difference in the varroa population between the parental and the daughter and the control colonies. So, okay, this is promising. But it's also important to look at brood numbers too. And I don't have the honey production numbers here, but my fear as a honey producer is I'm decreasing my adult bees, which are my foragers, to take advantage of the nectar flow that's occurring in May and June. So let me explain this slide, and I'll tell you how I dealt with that problem. If we look at this brood, again, we see that by September and October, there's no significant difference in the amount of brood between the controls and the treatments. So that was important to me as well. But this honey production really bothered me. And we did see a decrease in the amount of honey. The control colonies did bring in more honey. So I was like, oh, because beekeepers aren't going to like that, right? And so I thought to myself, what if we look at a later split? What if we don't split till after the main nectar flow? Brilliant, right? Well, I don't have the results to show you yet because we just did that last year, or this last season. But we have really good survivability. And this time I split into nukes for overwintering. So I actually reduced the size of the overwintering unit. So this is ongoing work, but overall, you know, what's really interesting, if we look at the population averages from these years, you can see the bees here, adult bees, varroa numbers here, and the brood, and these are control, parent, and daughter. Sorry, it's kind of confusing. It's not the best way for me to show this. And these are the months. And if you just take, say, the varroa and compare them across these, going across the months, you can see here the differences between the ones that were split and the control colonies. What's interesting, though, is then in October, a month later, you see this leveling out. And so really, why we're getting such weird values for the mites, some of these values are really hard to interpret properly, is because a lot of times at this point, there's not a whole lot of brood, so we're looking at only phoretic mite drops. So we really need to come up with a way to use the reproductive mite numbers, the ones in the cells, in the brood, to understand infectivity rates. So can we use splitting as a tool for mite reduction? Yes. The next question is, when's the best time to split? And we're working on it. We do a lot of outreach because we want to give beekeepers in our area all the tools they can. So a lot of times we're just trying things and we share it with others and we hope for them to try it. And so we designed this begonia, pretty clever, huh? And we made this really cute little bug go around. My student did this, and I promised her I would show this slide because she spent a lot of little time making this. That's pretty cute, huh? The little bee driving it. And we call it research by the beekeeper for the beekeeper. Because we need to learn from our colonies, and we can't put all of the research on, universities, because we know a lot. We can do a lot of research ourselves and increase our research numbers. These are our begonia participants, and this actually is not just for the projects I showed you. We actually had a large start off. People had over 100 people, and now we're down to about 50. So there's always a real excitement at the front end, and then as we're like, where's your data? They're like, oh, I don't want to do it anymore. But with our nematode project we did for small hive beetle, we had 600 to begin with, and close to 300 that stayed in the project. Wonderful. And it's because we gave them nematodes. If you give a little something, then you'll get better participation. But anyway. And this is the state breakdown from Alabama to Arizona, Arkansas, California. You can see just really, really great recruitment. And we also did a survey, tried to collect data from the participants, just seeing what their knowledge of IPM was, to see what their biggest fears, what their biggest challenges were in their apiary. Also to look at the breakdown. About 84% of the respondents were hobbyists. About 80 had less than 10 colonies, 83 less than 10 years. 80 didn't know why their colonies died if they had died or dead colonies. And so our future ambitions are to attain a better understanding of the relationship between varroa mites and their effects on honeybees, and how we can use simple management tools that we use all the time to create a dynamic apiary. Maybe we have some hives we split, some hives we don't split and we control, so that we can produce honey. Do we have to do the same thing to every hive every year? So trying to understand how to have this sustainable dynamic apiary. To end it, I'd like to just give the definition of sustainable. And it's able to be maintained at a certain rate or level. Okay? Conserving an ecological balance by avoiding depletion of natural resources. So I think when I read this definition, it really speaks to why and how I want to run my apiary. All right, so why sustainable and why splitting, or the common term increasing every year? It helps manage pests and pathogen loads, helps manage populations, helps in maintaining young queens. How many of you, when you go into spring, have this huge colony and you know it's going to swarm? You're like, how do I keep it? Splitting is a good option. Helps maintain a certain number of colonies and increases the odds of survival. If you split going into winter, then you've increased the odds of survival of colonies coming out of winter. So going back to nature, learning how honeybees do it without us kind of bumping into them. And also, this is essential in making sure that our bees have a food variety in order to live off of. And I want to acknowledge all the people that helped make this work. It was funded by the Environmental Protection Agency, and also the University of Delaware and Penn State partnered on some of it. And the Chester County Beekeepers Association were just awesome, really great group. And see, she puts bees on everything, sorry. A special thanks to the National Honey Show again and all the sponsors. I believe the BBKA is sponsoring this particular lecture. And with that, I probably went over, and I would be happy to take any questions.
Do you have any particular views on queen caging, isolating the queen in the hive?
The question was, do I have any views on queen caging, isolating the queen, kind of breaking the cycle that way? Is that right? I don't. I think it's a good idea. I haven't done my break in the brood cycle that way, but I think the key is the break in the brood cycle. I guess what I would wonder about doing that is, does that have any physiological effect on the queen to stop her from laying and then start her to lay again? I don't know. I know that a lot of people are doing it.
You said you had two apiaries, one which was a splitting apiary and another which was a swarming apiary. Did you get better results with the swarming apiary?
Great question. I knew somebody was going to pick that out. The swarming data is trickier because the hives in that apiary are from different years. So the splitting apiary was all first-year hives. And you know the difference between a first-year and a second-year hive. It's tremendous. In the swarming apiary, there's a lot of noise. So we're still analyzing the data using regression, some other analyses that are better at kind of reducing the noise, which is the fact that some are first year, some are second year, some are third year. But overall, we had better survivability overwintering in the yard where we tracked swarms. And so that is as much as I can divulge at this point on the swarming yard that is true, that I know is true.
When varroa first came to this country about 25 years ago, it was suggested that possibly migratory beekeepers had less varroa because the heat generated by the bees in transit actually destroyed some of the varroa. Have you found anything of that nature?
So he was asking about when varroa first came, the migratory beekeepers, in the movement of bees, it generated a lot of heat, and that actually killed varroa. And have I heard of that, or do I see that? Or is that the question? I've heard of that, but I know of no research that I've read that's really looked at that in terms of the migratory beekeeping. Most of the migratory beekeeping done in the U.S. is looking at like creating these crazy temperaturized trucks, and then looking at immune genes and seeing if they're being turned on or expressed. I don't know of any work that's actually looking at heat. That's a really interesting question.
I'm a new beekeeper and I'm hoping to use shook swarming, so no chemicals to hit my varroa populations. You haven't mentioned that. Is it because you don't think it works? Do you have a view on that?
Shook swarming. So
Well,
explain to me what shook swarming, what you consider shook swarming here.
What I learned and have tried is in April to basically shake your bees out of your brood boxes into new comb and in a new brood box, keeping a frame or 2 frames of uncapped brood in there, so that the mites on the bees you've shaken in the new box all go to those frames. And then after 3 days you throw those 2 frames away and destroy them.
Did everyone hear that? Okay.
And if I've got that wrong, I'm sorry. I'm
No,
new
no,
to.
that sounds. And you know, we didn't look at shooks. We looked at more classical splitting because that is a technique most of our beekeepers are familiar with. That sounds just like a wonderful way to do it. But we have not looked at shook swarming. Thank you.
Thanks very
Thank you
much,
so
Deborah.
much. Thanks.